Welcome children. Today's class is about cell reproduction. Every cell during its lifespan goes through a series of changes. Let's study the cell cycle. Every cell passes through two major phases, an interphase or I phase, mitotic phase or the M phase. The interphase is the longest and has the following phases in it. G1 phase, also called as first growth phase or post-mitotic phase, S phase or the synthetic phase, G2 phase or the second growth phase or post-synthetic or pre-mitotic phase. A G0 phase is also present, which is also called as a gap 0 phase. Let's try to understand what happens during these different phases of the interphase stage, which is also called as the preparatory phase, as it makes the cell ready for the cell division. For example, mitosis. During G1 phase, proteins needed for DNA replication are produced. There is also an increase in the number of cell organelles and also in an increase in the volume of cells. The cell, if it is non-replicating, for example, neurons, RBCs, skeletal muscles, will not move through the S phase and the G2 phase and the M phase or the mitotic phase. From the G1 phase, it directly enters the G0 or the gap 0 phase. During the S or the synthetic phase, chromosomes replicate, which means chromatids are formed, but the centromere does not split. Hence, there is no change in the ploidy of the cell. During the G2 phase, the proteins needed for cell division, that is, for the formation of asters and spindle fibers, are formed. Also, RNA synthesis takes place. The cell division actually means production of new cells from parent cells. There are three types, of which amitosis or direct cell division is mentioned first. It was reported by Remarque in 1841, observed in bacteria, cyanobacteria, yeast, amoeba, etc. In such kind of cell division, there is no presence of spindle formation to be seen. A single cell with cytoplasm and nucleus constricts, that is the cytoplasm as well as the nucleus constricts, it elongates, and two daughter nuclei are being formed, and the cytoplasm too divides to produce two daughter cells. This kind of cell division is referred to as amitosis or direct cell division. The second type of cell division is mitosis, also called as somatic cell division, as it takes place in somatic or body cells. It's called equational or homotypic division, as the number of chromosomes remain the same when compared to the parent cell, and qualitatively too the daughter cell is similar to the parent cell. Mitosis was observed in plant cells by Strasburger in 1875, and in animal cells it was observed by Walter Fleming in 1882. In the plant cells, mitosis occurs, or in plants, mitosis occurs in the meristematic regions, that is the root and shoot apices, intercalary and lateral meristems. Whereas in animals, it takes place in the somatic cells of the developing embryo, and in adults, it is seen in the skin cells and the cells of bone marrow, and also in damaged cells that need cell replacement. Let's try to understand what mitosis is. The first series of changes come under karyokinesis, which means division of nucleus. Prophase is the first among it. Can be divided into early, middle and late. It is an animal cell and centrioles can be seen. The first change that takes place is the centriole pair moves to opposite poles. Chromatin thread coils, shortens and thickens to form chromosomes, as you can see here. Asters are being produced by the centrioles. Further shortening and thickening of chromosomes take place in the middle prophase. The nuclear membrane and the nucleolus begin to disappear during this phase. By the end of prophase, we happen to see such changes: the complete disappearance of nuclear membrane, nucleolus, microtubules or bundles are being formed between the two asters. The spindles formed and the asters collectively is referred to as the mitotic apparatus. Next comes the metaphase. During metaphase, the chromosome looks like this with chromatids. In the center, we can see a centromere, a constriction, with a small disc-like structure attached to it called kinetochore, having 3 layers, outer, middle and inner. It is to the kinetochore that the spindle fibers are being attached. The spindle fibers attached helps in the movement of chromosomes. During metaphase, the major change that can be seen is the centromere of the chromosomes align on the equator of the spindle called metaphase plate or equatorial plate. As we can see, all the centromeres align on the equatorial plate. Continuous spindle fibers run from one end of the one pole to the other. Asters also can be seen. During anaphase, which is the shortest of the phases of mitosis, the centromere of the chromosomes split and the centromere points towards the poles and it gets pulled by the spindle fibers towards the poles. So the sister chromatids or the daughter chromosomes are either in the V shape as seen in this picture, J, L or I shaped. As in the previous phase, asters, continuous spindle fibers, tactile spindle fibers, that is the one attached to the centromere, are also seen. In addition to it, there can also be the presence of fibers in between the chromatids called as the interzonal fibers. Next is the telophase, where the chromosomes reach the opposite poles and they form two groups. On either poles, they uncoil and form the chromatin thread. Mitotic apparatus disappears, that is the spindle and the asters disappear. The nuclear membrane nucleolus appear. Immediately after that, division of cytoplasm, also called as cytokinesis, takes place. In animal cells, it is by the formation of a constriction or a furrow which develops towards the inside and splits the cell into two. In plant cells, because of the presence of rigid cell wall, furrow is not formed. Instead, the vesicles of endoplasmic reticulum and Golgi complex produce cell plates which are laid in the center, which later on develop towards the sides and split the cytoplasm into two. The stages of mitosis are same in plant cells, with the exception that asters and spindle fibers are absent, and during cytokinesis there is cell plate formation. All other phases appear the same. Importance of mitosis. It's important for the growth and development of an organism. It maintains the cell size, that is, prevents a cell from becoming unusually large. It also maintains the chromosome number of a cell. It is needed for the repair, regeneration and healing of a cell or an organism. It's also important in asexual and sexual reproduction. All the different phases of mitosis are similar in all organisms. Hence, it is an evidence of the basic relationship between organisms. And last, colchicine is a mitotic poison which is obtained from the plant Colchicum autumnale from the family Liliaceae. The presence of colchicine in cells inhibits the formation of spindle fibers and hence inhibits cell division. That's all. Thank you.